So in this video, we're going to look at microscopes and also the process of magnification. So first of all, before we think about microscopes, we need to go over two terms that are really important, which are resolution and magnification. I'm going to start with magnification because that's the one you've definitely come across before. Magnification is how many times larger an image appears than it compared to its real size. Now, that could be an image as in when you look down a microscope, or it could be an image that's on an actual drawing. It doesn't really matter. The idea is how many times larger is it, or smaller, but normally we're thinking about larger. And magnification is really useful, but magnification on its own is not very helpful. You also need the idea of resolution. So for example, if I took a picture and I blow it up, sometimes the picture will still be really, really clear when it's blown up, but often it will become blurry, and that's because its resolution is quite poor. So having a big magnification with what we call a low resolution is not very helpful. Now, what do we mean by resolution? Now, resolution in some ways is very similar to when we're talking about sort of pixels in a camera. It gives you the ability to see a detailed object, to really see the small details. We define it as the ability to distinguish between 2 points that are close together. And the higher the resolution, i.e. the more able you are to distinguish those between those 2 points, the more detail you can see. So if you want to look inside a cell and look at the small organelles that make up it, you need to actually be able to see things in lots of detail. You need to have a really high resolution. So when we think about our microscopes, we need to think about those sorts of things. So in terms of the microscopes, you're going to come across three that you need to know. The light microscope, which is the microscope that you're used to in school, normally plugs into the electricity and shines a light through some lenses. You've then got two electron microscopes. You've got the transmission electron microscope. You'll be pleased to know that we abbreviate that to a TEM. And a scanning electron microscope, which is an SEM. Both these things are too big for us to have in schools. They do have them in universities and in research labs. Now, if we're going to compare them, it's easiest to look at this table. Okay, so it starts by looking at what the actual microscopes do. So if we have a look at the light microscope, as we know, it uses light and lenses to focus an image. The idea being that you've got your microscope, it's got a lens in the top, an eyepiece lens. You then come down, and then you have three other lenses called the objective lens. And then underneath that, you have the stage in which you put the slide. And you're going to have a light source. And the light shines up through the stage, through the lenses, through the eyepiece lens and into your eye. And you obviously are going to focus that using the light, using the lens, and allow you to see the image. Now, the thing with light is light microscopes have quite a low resolution. And the reason for that is that light itself has quite a long wavelength. So therefore it becomes really difficult to distinguish between points that are close together. It also has relatively low magnification, really useful in school, but not much else. But the good things is it does allow you to see living things, and it does allow you to see things in colour. So you will have used it to look at sort of onion cells. You can use it to look at slices of muscle. You can look at little Daphnia water fleas, and you can see them alive and see their heart beating, and that's okay. But if you look down a light microscope and you look at something like an onion cell, you're not going to see very much. All you're going to see is the nucleus, the cell membrane, and the cytoplasm. You're not going to see anything else. So it's good for looking at some things, but not great for looking at others. So the second two are electron microscopes. Now, electron microscopes, as they suggest, use electrons to make an image. So in the case of a transmission electron microscope, the electrons are either absorbed or transmitted by the actual specimen, and then that is recorded below. In the case of a scanning electron microscope, the electrons are reflected back, and then that image is recorded. Now, in terms of magnification, they have a much higher magnification than the light microscopes. The highest one will be the TEM, slightly lower will be the SEM, but still much higher than the light microscopes. And really important is they have a very high resolution, and that's because electrons have a short wavelength. And notice how I say electrons have a short wavelength. That's really important. They often ask you in exam questions, why is a TEM used and not a light microscope? And you need to say, because the TEM has a high resolution as electrons have a short wavelength, or vice versa for the light. The highest resolution is for the TEM, then the SEM, and then we've already said that light has the lowest. The problem with the TEM and SEM is that you have to do them in a vacuum, and therefore they can't be alive, they have to be dead. And in the case of a TEM only, you will take a very, very thin sample. So what is the difference between a TEM and an SEM? Well, a TEM is to look inside the cell. You take you will take a sample and you will slice it very thinly with a really sharp blade. And you get a very thin slice, and then you can look inside what's going on. So in the case of a cell, we can start to distinguish the other organelles, lysosomes, the Golgi bodies, the SER, etc. You can start to see detail. So if that's the case, why have the scanning? Well, the purpose of the scanning is a slightly different thing. The purpose of the scanning, and as its name suggests, we said that you shine electrons and they bounce off. So the purpose of the scanning is to look at a three-dimensional image of the outside of a cell or organism. So all those pictures that you see online, which show you the red blood cells with the biconcave shapes in 3 dimensions, they're all taken with an SEM. So if you see a picture in 3 dimensions, you know it's a scanning electron microscope. If you see an image in 2 dimensions, it must be a light or a TEM. If you can see detail inside the cell, it's a TEM. If you can't see any detail at all, then it's going to be the light. Both electron microscopes are in black and white, although the SEM pictures are often coloured in after the event. The original image was in black and white. This is a really nice little table that you can use. You're often asked to compare light to electron microscopes, so it gives the differences between them. So this allows you to very clearly look at the differences between them. So the last thing I want to look at in this particular video is the idea of the magnification calculation. Now, you've come across that at GCSE with the triangle IAM, where magnification equals image size over actual size. Now, the important thing at A level is we're going to start to play around quite a lot with the units. You need to be able to convert your units from one to another. So we know that we deal with meters, millimeters, micrometers, nanometers. We can go as far as picometers, but we tend not to. So to go from meters to millimeters, you're going to multiply by 1,000, because there's 1,000 millimeters in a meter. There's 1,000 micrometers in a millimeter. There's 1,000 nanometers in a micrometer. So if we want to go backwards, we do the exact opposite. We divide by 1,000, divide by 1,000 again, divide by 1,000 again. So it's really, really clear. So it's 1,000 millimeters equals 1 meter. 1,000 micrometers equals 1 millimeter. 1,000 nanometers equals 1 micrometer. And you will notice I've left out centimeters. It's because it's not an SI unit. It doesn't fit into this pattern. If you're given something in centimeters, convert it into millimeters first. You should know how to convert from centimeters to millimeters very easily. So always convert it into centimeters. So if you get given something with which is 1.9 centimeters, right, that's 19 millimeters, which is therefore 19,000 micrometers, which is 19 million nanometers. Now, why is that important? Because they will give you things in wrong order. So, for example, the first type of question seems relatively simple. I give you a picture of a sperm cell, and I say that the actual size is 4.3 micrometers, and I want you to calculate the magnification. So the first thing you want to do is you're going to take your ruler. So you're going to take your ruler and you're going to measure the length from the beginning of the sperm to the end of the sperm, but I want you to measure it in millimeters, remember, our SI unit. So let's imagine that we measured from there to there, and it measures 22 millimeters, sorry, 220 millimeters, say 220 millimeters. So we now know that our sperm image is 220 millimeters, which we know is 220,000 micrometers. Why do I convert into micrometers? I convert into micrometers because this actual size is in micrometers. I then can fill in my equation, 220,000 divided by 4.3. Put it into your calculator. Nobody expects you to know calculations like this off by heart. You are allowed a calculator, so use it. And it gives me 51,162. So I know that my sperm is drawn just over 51,000 times bigger than it is in real life. So that is the first of the two calculations. They give you a picture, they give you the actual size, or they give you the magnification, and then from there you have to do a calculation. Always remember, measure in millimeters, then convert into micrometers. Sometimes they give it to you slightly differently. So for example, maybe I'm going to draw a cell. I'm not going to draw all of the organelles in. So like that. And I'll have A to B. And then I'll have a little scale bar here. And I'll say that scale bar is 4 micrometers. And I will say that to calculate the magnification of the cell. That's possibility number 1. Now, at this point, people often do really complicated things, but actually, you don't need to. They've given you a scale bar here for a reason. It's like a scale bar on a map. It tells you that from there to there, in real life, is only 4 micrometers, but in this picture, is whatever that distance is. So let's imagine that distance, measured in millimeters, of course, is 30 millimeters. The magnification of this scale bar is identical to the magnification of the cell. I can ignore the cell completely. All I need to do is say, well, 30 millimeters is 30,000 micrometers. Just need to divide that by 4. That will tell me the magnification of the cell. In other words, the cell has been magnified 7,500 times. So that's the first of the possibilities. Possibility number 2 is actually they want you to calculate the real size of the cell. Now we know that from there to there, in terms of our scale, is 4 micrometers. So the first, and we know it's 30 millimeters long. So the first thing we're going to do is measure from there to there. Now again, I'm just going to make up some lengths. Let's say that is, for ease purposes, 115 millimeters. So we know that 5 of those, if we do 150 divided by 30, we have 5. So we know that 5 scale bars equals 1 cell. And we know that each scale bar is 4 micrometers. So we know that 20 micrometers equals 1 cell. So that's the other type of calculation that you could get. So either they'll just give you, like I had before with my sperm, they'll give you a picture and give you the size and magnification, ask you to calculate something else, or they will start giving you scale bars and start asking you to work with those. We will look at the actual structure of the cell in a different